So the next group of really big molecules that we're going to be talking about are lipids. Now, I don't want to be misleading here. Lipids are not true polymers, okay? So we're not talking about putting individual little subunits, in other words, monomers, together to make them. But they do have a whole lot of bonds that can be used to store energy. So we'll talk about the structure in a second. Now, lipids, fats, right, um, have generally little or very low affinity for water. In other words, lipids are what we call nonpolar molecules, right? So they have nonpolar covalent bonds here. What that does is literally get water and lipids to repel each other, right? So they basically repel water. Now, if you think about it, if you've ever, poured olive oil or, whatever into pasta water, or Italian dressing where you have kind of like you have to add water and oil, they completely separate, right? And they'll stay separated. They're literally electrically repelling each other based on how the molecule is put together. So, important to consider. Biologically, what's important to consider is that most important lipids are fats, something called phospholipids, which we'll get into when we talk about cell structure, and steroids. Okay, steroids are really important in that they make a lot of hormones, right? Hormones being things like testosterone is a steroid, estrogen, which we more of like considered a female hormone is a steroid, although males and females have both types of steroids, right? Both types of hormones. Progesterone, another female hormone, but males have it too, right? So these really do play a really big role in body function. So to look at a fat, okay, fats are constructed from two types of smaller molecules: glycerol, which you see over here in the gray box, and a fatty acid, which you see over here in the orange box. Glycerol is a 3-carbon alcohol. And the reason that it's an alcohol is because it's got these OH groups on the side, okay? You don't really have to worry about that, but it is attached. See what's going on here? You have an H and an OH, and it gets kicked out as a water. So what's going on here? That's dehydration synthesis again, okay? So that's what attaches this fatty acid. See all these, C's and H's, right? CH, 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 that bond it to glycerol. Now notice that there are actually 3 places that have OHs. That means that there's 3 places that you can bond and another whole chain of these. Okay, so fats can actually be pretty large and have a lot of bonds to them. So fats separate from water because water molecules form hydrogen bonds with each other and exclude the fats. I know it sounds sad, right? But they just do not let the fats sort of like integrate in between the water molecules themselves. The long hydrocarbon chains of fatty acids can't form the bonds, and therefore they're hydrophobic. So that's what I mean by water repelling the fatty acid tails, the orange box that we saw earlier, from fats. In a true fat, the ones that we normally refer to as being important to life, 3 fatty acids, those long orange boxes, are joined to a single glycerol molecule by something called an ester linkage. You'll get into that in organic chemistry if you ever take it, creating what's called a triglyceride. Okay, triglyceride looks like this. Now you'll have to memorize this structure for your quiz. I'm totally kidding. Why would I do that to you, right? This structure here is a fat. Remember how I said the energy is in the bonds? Look at how many bonds there are here to break for energy, right? So that's why fat, out of all the things you can eat, right, has the most calories per gram, right? So this is a very, very high calorie substance that we and other animals can break down for energy. This is why also we love the taste of fat, right? Doesn't it make sense that we would really like the taste of what gave us the most energy, So things like butter, bacon, ice cream, all these really fatty foods are really, really good. They taste delicious. However, eating too many of these calories can obviously lead to problems. So types of fats. So fat structure, just like carbohydrate structure, can vary. So fatty acids vary in length. So in other words, the number of carbons in that long chain and in the number of locations of double bonds in the chain. That's going to be important because that's going to actually have a effect on the shape of the fat molecule itself. Okay. Saturated fatty acids. So you may have heard of saturated versus unsaturated fat. Okay. Saturated fatty acids have the maximum number of hydrogen atoms possible and no double bonds. I would just really kind of like focus on this because I think it's hard to think about all those hydrogens. Saturated fatty acids therefore have no double bonds. Okay. Unsaturated fatty acids have at least one or more double bonds. Okay. And I'll show you what that does to the structure in a second. Okay. The major function of fats in living things is energy storage. Humans generally keep fat around our organs, in our liver sometimes, and under our skin. Okay, so obviously, other animals can keep it in other places. If you find a fish or something like that, fish generally keep fat in their livers. So they don't really have any, fat underneath the skin on the outside. You ever see like, a fish that had love handles? I never did. So that's just not going to happen. Fats made from saturated fatty acids obviously are called saturated fats, and most animal fats are saturated. Okay, I would think of this as basically, how you tell the difference, in practical aspect would be what happens to them at room temperature. Saturated fats are solid at room temperature. So that would be things like bacon, right? You can take bacon out and yeah, it'll get a little bit soft, but it'll stay solid at room temperature. It's not until you throw it on that hot griddle that the fat melts and, forms a liquid. So the problem with that is a diet that is rich in saturated fats can contribute to fat depositing in places where you really don't want it to deposit, like in your arteries and veins, right? If that happens, that makes it harder for your heart to pump blood around your body, and obviously that's not good. Okay, so saturated fats not so great for you. Okay, these are all fats that have, remember, no double bonds. So those tails, that orange box, and you can see it's still kind of here with all the hydrogens and carbons in it, is relatively straight. Okay. Um, and what that leads to, we could discuss it in class, but what that leads to is staying solid or relatively solid at room temperature. Butter is going to get softer, but it's not going to just spread all over the place, at 70 degrees in your house. Now, what will, of course, is oils. Okay, so unsaturated fats, those fats that have at least one double bond in their tails, are generally liquid at room temperature. So you can see that there's a bend in this molecule, right? So I'm just going to say bent because I said straight in the other one. This bend in the molecule is what leads to it being liquid at room temperature. And again, we can discuss all that physical stuff in class. Plant fats, olive oil, corn oil, canola oil, vegetable oil, any of the oils and stuff are obviously liquids. Fish fats, which is interesting, are also unsaturated. Okay, so this is why they say, eating fish, which has a very, very small amount of fat in the actual, meat on the fish, right? Are generally better for you than saturated fats. So, you may have heard of like the Mediterranean diet, really high olive oil consumption, which is a good fat, avocados, and, fish, right? Very lean protein. So, a little bit better for you than than the saturated stuff. Okay, so phospholipids. These are kind of a little, sort of offshoot of fat, and instead of being bound solely to a glycerol molecule, as you can see here, they're bound to a phosphate that, if you're looking at it, is PO4, and choline, which is this molecule up here. So what a phospholipid does is it forms your cell membranes. You have, I can't even imagine how many molecules of this substance you have in your body, which basically keeps the good stuff in your cells and the other stuff out, right? So this is a phospholipid. We're going to be talking about this a lot, and usually they're represented by this little thing. Kind of looks like a balloon with two strings, You have a hydrophilic head, so this loves water, right? And hydrophobic tails, which actually don't like water very much. So when phospholipids in large numbers are added to water, they self-assemble. This is kind of a weird property into a bilayer. Okay, so in other words, and I'll show you a picture in a second, they actually form their own structures on their own. No life really is necessary. If you put phospholipids in water, they'll do this by themselves. Okay, they also may assemble into what's called a mycel. Ever use micellar water, to on your face or whatever? A mycel is a little spherical, oh, there's an H missing there, a little spherical shaped arrangement of phospholipids where the tails all point into the center of the sphere. So what I'm saying is all these phospholipids will form, it's not a bad circle, right? A circle, okay? Um, the structure of phospholipids therefore represents basically the same thing you're going to find in cell membranes. And if you look at this, wow, holy crap, look at all those phospholipids, right? And this is just around one cell. That's the head. I'll just use this one. And these are the two tails that are coming out of it. So this is the hydrophilic part. That's the phosphate head, along with the choline on the top. And then the orange part here is the fatty acid tail. In other words, it's the lipid. Okay, so we call this, what you're seeing here, since it's two layers of these, right? One's upside down to the other one. We call this a phospholipid bilayer, right? So there's a bilayer, there's two layers, and there's phosphate involved and lipids involved, right? So that's why we call it that. This is what the structure of every one of the cells in your body, blood cells, bone cells, nerve cells, fat cells, all have to keep stuff in the environment that should be maintained in the environment and out of the environment, or sorry, out in and out of the cells, really, I guess what I'm trying to say. So this is how the cell regulates what goes in and out of itself. Okay. Now the last class that I want to talk about as far as fats go are steroids. Okay, steroids are lipids. They have that hydrocarbon tail. That's what this thing is over here. Okay, that are characterized by a carbon skeleton that has 4 fused rings in it. Okay, so this would be like 1, 2, 3, 4. These 4 rings can vary, and that'll determine what these steroid compounds do. Cholesterol is also part of your cell membranes. It actually helps to keep things moving through and within your cell membranes. Um, so it's really essential, but obviously having too much of it is bad, and I think we've all heard that before. So before I move into proteins, oh, sorry, I thought there was one more slide for steroids here, but before I move into proteins, I think I'm going to end it here. This is only about going to be like a 13-minute presentation. Um, so let me just highlight, if you have any questions, I know we're getting a little bit more complicated in the chemistry. Don't get bogged down in the chemistry and all that stuff, right? I mean, you should know the general basic stuff, but you don't have to remember structures or, even chemical names other than the ones that I've highlighted, right? You don't need to know the structural difference between like glucose and maltose or something. Um, the theory behind it is really what I want you to pay attention on. Okay, so if you have any questions or you're unsure about something or whatever on the review for this unit, obviously you know where to find me. Hit me up on Remind, or please come to one of those meetings that I have, twice a week, and we can talk about it.